Susie,
Rob.
you've been studying dinosaurs for a long time. Did you ever stop and think about what they were eating?
Well, of course. I mean, my dinosaurs are primarily herbivores. We can tell that by the shapes of their teeth. But of course, what they might have been eating could have changed or did change through their evolution because they were around for quite a long time, Rob. And, at first, probably things like horsetails and ferns and stuff like that. But then, of course, angiosperms, flowering plants, evolved. Well, when did they evolve? Like maybe the early Cretaceous, perhaps even the late Jurassic.
Well,
And so
actually,
they could have.
that's been a mystery. Yeah, that's been a mystery going on ever since the time of Darwin, and that's exactly what we're going to get into today. When the flowering plants, the ones that you and I like to eat, you especially because you're the vegetarian, right?
More or
Yeah,
less, yes. Yes, indeed.
more
Well,
or
that's
less. Oh,
exciting.
yeah. So we're going to find out all about where plants came from and what dinosaurs were eating. Susie, you and I haven't caught up. This is ages since we last caught up. What have you been up to?
I've been on holiday. I guess we haven't caught up because it's the summer, isn't it? It's
Yeah,
August
yeah,
and it's carnage because we both have children, and that makes
yeah.
summer carnage. I've been in France, in the French Alps. Been doing some mountain
Nice,
biking, some whitewater
nice.
rafting, some hiking, trail running,
Wow.
stuff like that. No rest.
How very exciting and Red Bull adventure of you.
How about you?
Uh, I've also been trying to manage various different kids' activity clubs, grandparents, baby play dates. Although they're not play dates anymore; they have to be hangs.
Yeah,
They're
hangs,
hangs now.
hangs.
Play
Yet.
dates is definitely babyish. We went to Spain to go and see the clips.
Oh, cool. Yes, of course you did. I saw your tweet.
It was amazing. I really was quite impressed by that. So it was 26 years ago that I tried to go and see it in Cornwall and failed.
Did
So
you?
it was.
You did. Why did you fail? What happened?
Well, it was cloudy in Cornwall, but we had a great time in Spain.
Oh,
But it was so hot. And then I found out that when we were there, it was 34 degrees. It was too hot to do anything, like other than go to the pool and get out again. And then I found out when I got back, Leicester was hotter. So, well done to the people of Leicester and Oxford who may have had to survive that.
I actually went to Cornwall to see the eclipse in
Yeah,
1999, I think it was.
yeah.
And I did see it. It wasn't cloudy where I was. I got to see
Oh,
the eclipse.
I actually was in Devon, so that might have been the thing. But
Um,
I said Cornwall because that's where it was most famously. So the thing is, the next one's not. I was talking to the kids about this. The next one in the UK is not till 2090, and we were like trying to weather up whether I may or may not be there. On balance,
Probably not. Probably not.
unlikely, right? They might, but
Yeah.
it ain't happening for me.
No.
Anyway, let's rather than thinking about our own mortality, let's think about flowering plants and angiosperms. So this is our first time doing a proper plant episode. We've never done that before.
No.
And I feel like that's a bit of an omission because we've done 40 episodes, two on fungi, which have virtually no fossil record, and this is our first time talking about plants.
Well, there was that large phallic, what was it called? Prototaxites, which is
Yeah,
that, were you counting that as fungi? Because it's not fungi though, is it, Rob?
well, it's not.
I think you're fine.
Okay, it wasn't a fungi, it wasn't a plant. You're right, good point. I was also thinking of our fungi, our brain bursting out
Yeah, that was a good one.
amber, the fungi taking over the brains
Yeah,
of ants in the
that
Cretaceous.
was a good one.
Bonkers.
Hmm.
But bona fide plants, no, nothing. So this time I want to talk about angiosperms and their origin. So this has been a problem for a long time, right? As you alluded to in the little bit at the beginning. Darwin was annoyed by this. He called it an abominable mystery because he was looking at the fossil record and he see the sudden appearance of lots and lots of different types of angiosperms in the Cretaceous sometime. But actually when they got going has been a bit of a mystery. Their phylogeny has been a mystery. So I guess we need to define what an angiosperm is. So that's the flowering plants, the ones that have, well, nice big juicy fruits and seeds, I guess, and flowers. I guess those are the two main things. But that's most of the plants that we eat are going to be belonging to that. I can't think of. Do we eat anything that isn't an angiosperm?
I can't think of anything. We eat seaweed. That's an alga.
Seaweed is not in that. Yeah, right. Okay. So apart from seaweed, I can't think of any gymnosperms. So ferns and
Mm.
are like a major type of plants we don't eat, but we do use them. And then there's lichens and mosses and things like that. But most of the plant diversity is concentrated in the angiosperms.
It's quite interesting, isn't it, Rob? Because the angiosperms are the things that really are colourful. And so
Yes.
before the angiosperms evolved, it must have been just, just fairly homogeneous and green. Terrestrial ecosystems must have looked quite different to today because they're just, sort of
Yeah,
green
that's a good
and no
point. I hadn't
colour,
thought about
no
the color.
no reds and pinks and purples and lilacs and, just
Well,
green.
the complexity of the ecosystems is something we're going to get into here in terms of like the canopies and the humidity and things like that, and the construction of modern forests. Like, what does the world look like? But also, grasses is an interesting one.
Well, yeah.
Like, they came along later. Like, when we think about what the world looks like in temperate environments, massive grasslands are an important component of that. But they didn't exist in the
No.
Cretaceous, really.
Not at all.
No, that was just something that came along later with mammals and plants and
Yeah.
all sorts of interactions.
I
Also,
mean,
the
much
interaction.
later though, right? I mean, like 30 million
Yeah.
years ago or something.
Well, it
Grass.
depends on how you measure it, but they may have originated. But in terms of widespread grasslands, yes, they came along quite later. Yeah, as an ecosystem. So it's quite hard to think about what a Cretaceous, vegetative landscape might have looked like. So this raises this question: how and when did these angiosperms get going? And that's what this paper we're going to talk about today: is diversification of angiosperm reproductive strategies predated the end Cretaceous extinction. And this was published in June 2026 in Science. So we're still pretty hot off the presses, although now it's August, I guess. Well, we need a holiday, right?
We need a holiday.
We love our fossil fans, but I need a break. Susie needs a break sometimes. So this is a smart gap. So Jamin Lee is in the lab of Cindy Liu, I don't know how to pronounce that, in Berkeley. So they're setting up this problem of what was going on with the angiosperms. When did they get going? So their dispersal ecology and their fruits they're widely thought to have mainly got going after the end Cretaceous extinction, when the dinosaurs went out, and mainly the seeds and the diaspores, which is a word I wasn't particularly familiar with, which is the seeds plus the structures and the fruit on top of that. So I think we might just use the word fruit from now on, even though fruit might not be technically accurate. We're talking about them being quite small throughout the Cretaceous and only got big later on. And they wanted to test that and have a look at it. But also, because this makes relevant, this is a massive difference because of the ecology, right? This isn't just a story about animals. This is a story about how the ecosystem was constructed and who was doing what and who was dispersing and how these things were happening. So it's a pretty big question. So in modern ecosystems, they also talk about how most things, the small diaspores, these small fruits, is associated with herbs, weeds, and that sort of. We're kind of imagining low-hanging vegetation, shrubby type things. That's what we imagine most of the angiosperms in the Cretaceous might have been like, on the basis of the fruits.
And how are their fruits being dispersed by animals?
Yeah, well, again, that depends.
Insects.
You have to look at the different types of fruit. Is going to depend upon the different kind of activity.
Okay.
It might be animals eating them. It might be wind dispersal. So you might have some sort of. That's why I maybe shouldn't use the word fruit, because you might have some sort of diaspore that enables you to be wind dispersal. If you think about a sycamore, the helicopter things, right? We've all done that. Yeah, that's a diaspore. That body around it.
Mm-hmm.
Okay, so that's the question. These team discovered a fossil carpoflora, so that's a fossil deposit composed of seeds, in the Lake Campanian, the Upper Cretaceous in New Mexico, and they're going to call it Dory's Tuft.
Are they? Dory's Tuft.
They need a better name for that.
They do.
Which is maybe why they abbreviated to DT throughout the manuscript, which, again, we've talked about this before. Don't put in unnecessary abbreviations. We can cope with it being Dory's Tuft. We can deal with that. So this is an inland floodplain environment, and it's a tropical forest. So I found in the supplementary information, this was kind of buried away, which I thought tells an interesting story, this one sentence. So, quote from the paper: the fossils for this study were collected in 1992, 1993, 1998, and 2012 by Dr. Garland Upchurch and Dr. Greg Mack and students, and in 2013 to 2016 by Dr. Dory Contreras and Dr. Cindy Liu and students. Thoughts?
Sounds like maybe they're doing undergraduate fieldwork there or something and collecting their fossils over a long period of time.
I was thinking more about the time frame.
Yeah, so they've been collected for 40 years, 30 years.
Yeah, they've been collecting this since 1992. So I presented SVP, and I felt bad because I presented a paper that first had its genesis in like 2007, and then I finally got around to presenting it nearly 20 years later. This is putting us to shade here. I mean, what's your longest project you've had in Genesis? How many fossils have you got in drawers that you're ready to work on?
Well, I tell you what, I think there's probably about 4 or 5, waiting to be described specimens in the basement right now. That I mean, my feeling about that, they've been there for a long time, but my feeling about them is that, well, they're not. They're not going anywhere. They're not going anywhere. They're not going to change, Rob, are they? They'll wait.
Well,
I don't have to work on a Saturday.
What if some visitor swoops in and snaffles them up.
Well, we tell them they're not allowed to do it because we're working on them. We're working on
Okay.
them.
We're working on them. Just give me another 10 years. Come on.
Yeah, exactly. So we have I think my personal longest running project was one actually a bit similar to you. So me and Richard Butler started working on some specimens of a dinosaur called Scutellosaurus, and we actually wrote a manuscript before we finished our PhD. So I think we had a manuscript written in about 2006. And we've
Oh
been to
no.
see some, but we hadn't seen these couple of specimens in Texas. And we felt like we didn't want to publish the manuscript without seeing them. And so, and then we just never saw them. We just never got around. And then I had a PhD student working on Thyreophorans, and so we were like, we should get him to finish it. And then he didn't really do it either. And then finally, in 2021, we actually worked with an American who had seen this material, and he finished the paper in 2021. So that was a really long running project. So these things happen, right? But I mean,
it can happen.
the thing with these, I'm going to put my hand up right now, bringing us back to today's paper. Like, I haven't read this paper, but it could be that they needed a critical mass of these things to be able. I mean, having found one, they might not have been able to publish it, right? They needed to find 50 of them to be able to, say what they, feel confident that what they were observing was right. And it just took them that long to find the numbers that they needed to feel confident about their interpretations, or to, get the full details of the fossils or something like that.
Absolutely. The significance of this paper lies in the fact that they were able to take a big sample through multiple field studies and get hundreds of specimens and then do a sort of ecological level study. It wasn't based on a handful. One or two specimens wouldn't have been able to answer the question they were dealing with. What they needed was a fairly comprehensive ecosystem analysis. So with that in mind, they've got 450 fossil diaspores. But they also have some leaves and some gymnosperms as well.
That's a lot of fossils. It's a good sample size, 450.
That's a good, yeah. And they've classified them into 77 morphotypes based on their shape, size, and texture, etc. 26 of those were fleshy. I shouldn't do inverted commas with my fingers in an audio format. So
I mean, it doesn't really work.
It doesn't work. 4 of which were winged, and 47 of them were other. So we have a lot of them, but we don't know what they are. So that is basically what we've got. We've got a large amount of these diaspores, 450 of them, and there are different types, and they tried to quantify the different types and estimate the size of them.
Okay.
So they did some maths based on looking at estimating the 3D volume of this thing, based on what they have found as the fossils. I mean, this raises an interesting question, because what happens to a fruit when it taphonomically, if you preserve it, right? If you've got a hard seed surrounded by some more fleshiness, and that might shrink or distort during taphonomy. I don't know,
totally.
but
you would think so, wouldn't you? Well, I was going to ask you, what is the taphonomic context of this? I mean, why are these? Because these things are going to be primarily soft bodies, right? Really, they're not. There's not much hard stuff to preserve in these, I guess. So, what's the taphonomy? What's the depositional setting? Why are we seeing all this plant material preserved?
So it's an inland floodplain environment. It's humid. It's a tropical forest potentially. But as for the actual question about taphonomy, they don't speak about that very much. And also, even though I'm the one that talks about, I profess to be the
taphonomist,
the taphonomist who knows about fossil preservation and all that. When it comes to plants,
got nothing.
I got nothing. So they say it's like, what they highlighted was that they have no reason to think that the taphonomy would have distorted the sizes here as in comparison to everywhere else, and I guess that's a fair point, right?
Yeah.
So I think that we'll allow that. Is there something specific about this where they might be more distorted or shrunk than anywhere else? I don't think they have any reason to think that. So that's fair enough.
No. Okay, fair enough.
So now we're getting to the headline. What they found out was that their carpoflora, the diaspores, the fruits and the seeds and whatnot here, were much, much larger than other Cretaceous things, orders of magnitude bigger. So the average size of these things was about a blueberry, which doesn't really strike me as massive, but it's still pretty small.
It's pretty small.
It's pretty small, but the average size of the other Cretaceous ones is about the size of a poppy seed.
Oh, right. Yeah, okay.
So tiny. If you think about how big a poppy seed is.
No melons then. Melons hadn't evolved yet.
There are some large ones. I mean, we have to bear in mind that a melon that you look at. This is the other thing that gets a bit distorted when you think about this. When we think about the size of the fruit, often we're thinking about things that we've artificially bred to be absolutely enormous and look ridiculous.
Yes.
I don't know if you've seen what a wild banana looks like, but it is, all seedy and very, very different from the weird banana freaks that we still. Side note. Bananas might go extinct because they're all just like one tiny. Did you know about this?
No.
So
Why might bananas go extinct?
because of the selective breeding,
Yeah.
almost all bananas that we eat come from one single cultivar. So there's very, very little genetic diversity in bananas.
Oh
So if a
dear.
disease was to ravage them, we might end up,
That's it.
we're less likely to be a problem for things like apples because there's quite a lot of genetic diversity in apples. But bananas, forget about it.
Huh. Interesting.
Anyway, I obviously. This is what I have to be. I'm sweating now, not only because it's a bit humid and hot, because the rain has finally started happening in Manchester after
Has
weeks.
it? Congratulations.
Yeah, we actually have some rain here, so it's quite humid. But also, I'm sweating because I'm getting nervous talking with any confidence about plants. I've chosen a plant topic, and this. What do I know? But
on the plus side, Rob, no one actually listens to our podcast, so it doesn't matter.
That's not true. That's not true. They're out there. Thank you, our fans. Also, don't forget that time we were number 1 in Earth Sciences in the Czech Republic.
Oh yeah. How could I? How could I forget?
Yeah. So we've got a big size range. So they're not just big. We've got some big ones. The average is about a blueberry, but we've got some bigger ones.
Mm-hmm.
Have you got a look at some of the pictures? Can you describe us Can you describe us the figures, the fossils?
Yeah. So the fossils. I mean, I guess this is why I was asking about how they're preserved, because the pictures, they sort of look a bit like they might be on a sandstone or something. And I just thought it was kind of an interesting thing. But anyway, they're sort of blobby. There's some blobby things that look like they maybe got some kind of carbonaceous films, and then there's kind of some round things with some stems. I mean, they're mainly blobby, aren't they? There's something that looks a bit like an apricot, but obviously it's not because it's much smaller.
Yeah.
But yeah, they're sort of round, overly blobby things. I mean, they're not. I've got to say, I don't know if I found these that I'd go, oh my God, seeds. These are fruits and seeds. I'd be like, yeah, it's a smudge on a rock. But
Yeah. I wonder if the plant paleontologists say the same thing about the fossils that we look at. Like, I wouldn't have recognised that. But I guess with animals, you've often got a nice crisp bit of bone or shell or something.
Where you've got a bone-shaped bone.
Yeah, these are a bit more amorphous, blobby items.
Yeah,
they look like something, right?
They're definitely something. Yeah.
The apricot ones are quite compelling. Yeah, they look very apricot-y.
Yeah.
But what they actually might be, I can't really think of.
No.
Obviously, we both. I'm just for the listeners. Susie and I are both well aware that they're not apricots.
I mean, they're mainly kind of yellow. That's why they look like apricots.
So that's the sort of fossils they're dealing with, and then they've got a nice graph where they've described the diversity of the shapes of these through time. So all throughout the Cretaceous, they're remaining about 1 millimeter average, and then in the Paleogene and the Neogene, so after the big dinosaur extinction, we see a sort of orders of magnitude step change in the size of these. Now, with this new one from New Mexico, we can see that in the late Cretaceous, or upper Cretaceous, we see this new data point that indicates there's much, 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 much bigger size range than previously appreciated. So that's the new data point here. And that's what gets you a science paper, I guess, that one thing. So that's it. That's the data.
So I mean, it's interesting, Rob, because one has to wonder how much of this is a taphonomic artifact. I would have thought this size thing is we just haven't found good faunas. Have we got an equivalent kind of taphonomic setting? And of course, they haven't really gone into what taphonomic setting is. But I would quite like to see all of these different data points that they've got that kind of they're from the Cretaceous. In the early Cretaceous, obviously, we've only got 4 data points, and then as you go through into the later Cretaceous, you've got many more as angiosperms become more common and more easy to recognise, presumably in the fossil record as well. But it would be nice to know whether all of these are preserved in similar sorts of environments, or whether they're preserved in different kind of environments, because there could be some sort of environmental bias. That means that we're not preserving big ones, or we're not preserving small ones, or that this site is particularly unique as a Lagerstätte, whereas the others aren't, and that could be causing some kind of distorting the picture a bit. It's also interesting, isn't it, in the Paleogene, as you go into the Paleogene, there's some really quite large ones, and then the size seems to decrease, actually, back through the rest of the Paleogene and into the Neogene. So, again, is there some sort of taphonomic effect going on here? It's kind of interesting.
Yeah, I think you raised some really good points there. They do talk about the environments there. I mean, that's slightly difficult because the environments are changing all the time for us.
Right, exactly, as well.
Before. How do you detach the environmental change from what's going on with the ecological change, which is going on from the taphonomic change? That's quite hard. But they also said they have no reason to see that this one would be different, taxonomically speaking, than the others. But they did also highlight how one of the things that makes this difficult is that you need to systematically collect the data in order to do this. And you need to, you don't just want to be checking out the nice handful of big chunky ones, or you want to be looking specifically for the carpoflora. Now, I think that's one of the problems that we have with, one of the things I was thinking about this is that the systematic data collection of plants is a bit not happening in the same way that it was for dinosaurs, right? I don't know how many times that the plants are sort of shoveled out the way to get to the animals underneath, and especially not with thinking about whether or not you're collecting the small ones and the large ones to get the size range data.
Yeah, absolutely.
They highlighted that point because they say there are a handful of big ones in collections in North America in the Cretaceous, but what you really need to do this is a systematic collection. Anyway, I was looking at where they got this size data from, and most of it came from a paper from 1999, and I was kind of like shocked by that. I was thinking, if you did this study with dinosaurs, and you based it on things that have been known since only starting from 1999, think about all the things that have been discovered since 1999.
Probably about half of dinosaur diversity has been discovered since 1999.
That's the whole chunk. Now, I don't know if that's because plants are moving more slowly, and then I found out that they populated that themselves with some more data points that come along. It wasn't just the 1999 data, so I was less stressed out about that. But I do wonder now if paleobotany moves at a slower pace than paleozoology. I don't know.
Well, there's fewer people doing it, I suspect, isn't there? So I mean, there's going to be a sort of worker bias there.
Yeah.
So I would think
so. That's the data. Then we can start moving on into the speculation part.
We
enjoy a speculation, don't we?
Well, we do enjoy a speculation station, and I note that they've got a lovely picture, a lovely reconstruction.
I wanted to talk to you about that picture. So they have a paleo art reconstruction. Can you describe to us what this picture is?
Christ knows what the animal is supposed to be. I mean, it is obviously some sort of like. So what we're looking at is a picture where you've got some plants that are kind of they've got kind of leaves, kind of frond-like leaves, and you've got some mushrooms, and you've got a small mammal in the foreground. That might be a kind of Morganucodon type mammal thing, like a relatively small mammal. Obviously, Morganucodon is known from the Triassic, but we don't think that mammals got very diverse through most of the Mesozoic, right? So maybe they're just kind of still small.
It's some sort of multituberculate.
Oh, it's a multituberculate. There you go. Okay, all right.
And the
still an early mammal.
And some sort of marginocephalian is the thing in the background.
Is it a marginocephalian? So there's a dinosaur that's sort of poking its head through the leaves. It's presumably supposed to be a dinosaur. It's not totally clear that it's a marginocephalian. It has to be. It's got some quite elaborate eyebrows. They look like they've got some sort of, I don't know, kind of leaf-like structures on their eyes, and it's got a very knobbly head. So maybe it's supposed to be a pachycephalosaur.
What I really liked about it was that this is a really good use of paleo art to help you with your story, right? So we've got this like shady forest because it's quite high canopies potentially. We've got a variety of different seeds and plants and two different types of animals eating it, and then we've got some insects. This really tells the whole story of this paper in a really evocative way, in a beautiful bit of paleo art. So this is by Brian. I don't know how to pronounce his surname. I've met him. I can't pronounce his surname. Brian Eng. He's a nice guy.
He's a nice guy.
Also partner of one of the people that we featured, the scientists. So Yara Haridi, who we did I think they are, connected. I don't know. So he's a nice guy, and he's made some really beautiful art there. I think he's quite popular in dinosaur communities. But I wanted to ask you more about. So this is an example of how the paleo art is being used really well to tell the story, right?
One might even speculate, Rob, that the paleo art is actually, really helpful in getting this paper into science.
Well, potentially, we saw that with the giant octopus one, didn't we? Where you had this arresting image of a giant octopus. Well, I wanted to ask you more about paleo art more generally, in terms of adding it to scientific contributions. So paleo art is a big ongoing, important scientific and artistic endeavor, but often have you ever commissioned paleo art to go in your papers?
Yeah, lots of times actually. Yeah. And I think there's some good reasons to do it. Most of them are relatively cynical, I would say. I think that paleo art helps with kind of communication, doesn't it? It helps with public communication. And a thing is that if you want to do a press release on a paper, you really need a piece of paleo art, because that will guarantee you getting a media story on it. So you can do the paleo art, you can commission paleo art to accompany a paper and not have it in the paper, or you can accompany it to have in the paper as well. I don't know. I mean, actually, I would say that the one time where it was actually really useful in the paper itself was for Spicomelis. So I commissioned
Mm,
my former
mm,
student Matt Dempsey, who's a brilliant paleo artist,
mm-hmm,
to make Spicomelis reconstruction, and that was
mm-hmm,
actually really informative because we didn't know any of the bits went.
mm-hmm,
So he kind of used his knowledge of ornithischians, but also kind of where he could actually just physically fit the armor on the body to
mm,
actually, and it actually kind of helped us think about, what was the function of this armor and stuff. So it was actually
mm,
useful in
mm.
that context. Mostly, I'd say it's more just about making your paper look pretty, which helps to get it into higher impact publications. And whilst that
Yeah.
sounds cynical, hey, we're playing a
No,
game here, Rob.
we are, and I think this one is a good example because it's telling a whole picture. Not only is it visually appeasing, but it's telling a nice, interesting scientific picture. Your Spicomelis example is a really interesting one because that's actually forcing you to think about how the animal was constructed. I've only ever commissioned paleo art for a paper once where we were looking at bite marks in fossil fish, and we were inferring that they were being predated because we could see that the increasing bite marks through time. So I wanted an image of some big toothy, like lungfish type thing or stem tetrapod munching up a lovely
Mm.
innocent jawless fish. And so we worked with Giulio Lacerda. He was very nice and
Oh yeah.
kind, and we paid him for the art. And I remember feeling a bit grubby because the first version he sent was lovely, but I actually sent him back saying, "Can we have more blood?" I wanted it to look,
Well, there you
I wanted
go.
it to look more violent. So
Yeah.
I don't know if this is something that paleo artists are quite annoyed about. Like,
No.
oh, you want two animals fighting and looking dramatic again? Ugh. You don't want some beautiful vista over the Cretaceous sunset. No, blood.
Lad.
Anyway, there's no blood in this one, but the point is that they're eating the fruit. So now we'll get into the question of who's eating what and who's doing this, because this also relates to the question of dispersal. Quite a lot of these fruits at this New Mexican Cretaceous thing are fleshy, which indicates that they were being eaten and using animals for the dispersal. Who was doing the eating? So they highlight multituberculate mammals. So multituberculates is a large group of mammals. I think most of them went extinct if not all of them went extinct at the end
I
Cretaceous,
think they
because
did,
only
yeah.
a handful of mammals survived. But they had quite a big variety of their different diets and habitats. They also highlighted, now bad pronunciation coming, tapijarid pterosaurs were likely frugivorous apparently, and dinosaurs and birds were known to concern gymnosperm diaspores, but likely transitioned to angiosperm diaspores when they were available. Also, they may have looked at the coprolites and found dinosaur poo full of some of these fleshy fruits.
Mm.
So do you think so you've worked on lots of herbivorous Cretaceous dinosaurs, right? And
Yeah.
Jurassic ones, but you've worked on them. Have you ever reconstructed any of them eating fruit?
Oh no, we haven't. But I mean, I think it's completely plausible. I don't think
Mm-hmm.
why would they not? I mean, the plants. So there's been some work that was done, led by my colleague here at the NHM, Paul Barrett, on dinosaur and angiosperm co-evolution, and whether
Mm-hmm.
actually, uh, the evolution of angiosperms and dinosaurs went, hand in hand. As the dinosaurs
Mm-hmm.
started eating them, did they then,
Mm-hmm.
produce things like fruits which were delicious for the dinosaurs, so that they
Mm-hmm.
could be dispersed in that? And actually, they found in that work, they found that there wasn't any evidence for co-evolution between
Mm-hmm.
dinosaurs and plants. But I think a lack of evidence is different from it not happening. I mean, I think a lack of evidence could be due to the fossil record not being good enough to determine it, rather than because it didn't necessarily happen. But
Mm-hmm.
I mean, I think there's more evidence that that angiosperms evolved, things like flowers along
Definitely.
with insects.
Mm-hmm.
But I don't, if there are fleshy fruits at this time, surely they're for animals to eat them, aren't they? I mean,
Mm-hmm.
why else would a plant produce them? So the dinosaurs must have been eating them, because every animal larger than a meter in size throughout the Cretaceous was a
Yeah.
dinosaur. Oh well, except there's that big croc, which more or less was a
There were
dinosaur.
some big mammals, weren't there? A
No,
handful.
there was I think the largest size the mammals got to was about they were sort of badger sized.
Right. Well, there were also pterosaurs. So
Terrestrial animal. Okay, all right. It's a very vague stat.
I'm not trying to pick holes in what you're saying. I'm
Jesus.
just saying it's there are other things out there. But yes, pterosaurs, I guess, are not a million miles away from being a
Yeah, that's
dinosaur, are
true.
they?
Well, they're
So
close cousins, aren't they?
yeah. Well, they also talked about another thing. If you're not, so are you evolving a fruit to encourage a dispersal, right? So the idea is that you've got this fleshy thing. It's an idea that you might encourage an animal to eat you. Therefore, that animal's going to walk, I don't know, 16 miles away and then poop you out, and you're now find yourself in a much further away environment than you otherwise would do.
Yeah.
But fruits are not just for that. They also talked about potential exploding fruits. So like legumes. So they found some fossily ones that look like they might be the ones that sort of pop out, which isn't
nice.
about. That's a different
Okay.
way of dispersing.
Yeah.
And some legumes or some catkins as well. So that's kind of cool.
Okay, yeah.
So they infer all this story together about the interaction between the fleshy seeds and the consumption of the dispersal, but also that this is now a closed forest, a floodplain, not canopy. So they're not talking. They think it's a tropical forest, but not like a rainforest, because the canopy, a modern tropical rainforest would have quite a lot of wind dispersers, because the ones at the top are very, very tall, and therefore they will use wind dispersal. They didn't find very much wind dispersal, so they're saying it's not quite the full multi-tiered canopy rainforest, but we're on our way to getting a complex forest. So that's pretty cool. And they evoke some hypotheses about what's going on. One of them you've already touched on, which is the coevolution. What's going on between the coevolution between the frugivores, the things eating the fruit, and what the plants are doing. And then also maybe just as the plants got bigger, the fruits can get bigger. That's a more straightforward thing. Or recruitment. Maybe as you're changing from the environment to a more closed forest, and that you have to change the way that you're doing things, because wind dispersal is not as good in a closed forest. You have to then start doing different activities. So that was pretty cool. I think that's kind of fun. I mean, I don't know how much one data point. I say data point. It's not one data point, is it? It's one site.
450 data points, but it's one site. It's from one site. Yeah.
Can that overturn the general pattern of the Cretaceous ones being small? Because at this point, everything else discovered in the Cretaceous has been small, apart from this one site. I don't know. It doesn't necessarily overturn everything, but it's an interesting consideration, and it's enough to get you a science paper. So
apparently so.
Excellent. Yeah.
Yeah.
Good. So before we move on, any final concluding thoughts before we move on to the next planty angiosperm paper for you?
No, let's crack on. Okay. I'm excited for the next.
There was a second angiosperm paper, and potentially even a third I want to talk to you about. So this is all based on the diaspores, the fruits and the bodies. But actually, this is the thing that I find annoying about plants. You can't just look at, like with an animal, I can get a body and I can look at that body of the animal. If I've got a brachiopod or a trilobite or a dinosaur, I have a body. It's a nice consistent pattern. In the fossil record of plants, I have to deal with either the leaves or the fruits or the pollen. I can't deal with all those things at the same time because of the way that the taxonomy works. It's often a mess.
It's a bloody nightmare, actually, isn't it? And they have a completely different taxonomy than we do in zoology as well, as a consequence of that, right? So because, you might have found a leaf somewhere and go, oh, I'm going to name this as one taxon, and then a root and go, oh, this is another taxon. And then, you find the whole plant and you're like, oh, bollocks, they're part of the same plant. So they have these kind of concepts of sort of form taxa, which are like, a leaf. They're a bit like ichnotaxa, I think, aren't they? They're sort of a leaf taxon, and then a seed taxon and a root taxon, and they're acknowledged that they're not actual meaningfully, like biologically meaningful taxa sort of thing.
Yes, absolutely, which is one of the reasons they're a nightmare.
Yeah.
But I take my hat
pollen,
off to pollen.
When you get to pollen, like things produce pollen that all look like, there's lots of things that produce the same looking pollen as well, isn't there? So that's really problematic.
What a nice segue you're giving me, because the next one was going to be all about pollen. So this is by Yang Liao and colleagues from Kunming in China. Post-evolution shaped extant angiosperm pollen disparity. This is published in Nature Communications in 2026, January. So this one's a bit older, but it's sort of, I thought this is a nice partner to the other paper, because one was about the diaspores, the fruits, and this one's about the pollen. So it's a different kind of thing. Although I only realised after I got into it that it's not actually about the fossils, it's about the extant taxa. So less to say about that. So they've got, but the scale of what they've done is massive. So they've got 1,880 genera of angiosperms, modern ones, and they've got the pollen, and they've scored them for 20 characters, discrete characters. So that's a lot of work, right? And then they've tried to infer, using a phylogeny that they've built from molecular things, the timing and sort of disparity changes through time. So that's quite straightforward. So they've taken those, they've measured 1,880 pollen and scored each one of them for 20 characteristics. So when you get to that point, you can imagine the volume of data is enormous. So they've done a non-metric dimensional scaling, NMDS, or other people might use a PCA, a principal component analysis. I'm not going to explain all the maths behind it because people don't need that. But the point is, you'll have used these techniques, I'm sure. You take an enormous amount of data and you collapse it down to a sort of two-dimensional visualization of enabling you to make it more easy to work with. So they found there was differences between the eudicots and the monocots. I guess that's not massively surprising. Those are
No.
the two main divisions of angiosperms. I wasn't particularly shocked by that they might have different types of pollen. And then they tried to plot that through time in terms of the disparity. That's where it gets interesting. But so they see two massive pulses in increasing disparity. One in the middle of the Cretaceous, in the Albian Cenomanian, and then things get pretty steady throughout the rest of the Cretaceous. And then another massive pulse immediately following the end Cretaceous extinction. So that first one the second one makes a lot of sense, right? The end Cretaceous extinction, we're going to see global ecosystems completely disrupted. So we might expect some sort of level of innovation or change during that post-Cretaceous world. But that one during the middle Cretaceous is interesting. That implies that there's, a lot of diversity and activity going on in the time when do we think these angiosperms get going? They're saying that there's been two pulses of this.
Well, that time period is interesting, isn't it? In the sort of, as you say, the mid Cretaceous, which is an informal thing, because there are a number of hypothermals that occur at that time. So this is times in Earth's history where we've had very, very, very high temperatures. And the Cenomanian is one of them, I think. There's a Turonian one as well.
Oh, yeah.
And so there's a couple, there might even be three, I think, 2 or 3 in that time period. So it could be that these are, I don't know whether they talk about this in the paper, I haven't read it either, but whether these actually correspond, these diversification events correspond with these Cretaceous hypothermals.
That's an interesting question. They do include the Eocene in there. So the Paleo-Eocene thermal maximum is in their thing. So that's one of the that's one of the pulses. That's in the post Cretaceous. When I talked about that second pulse after the Cretaceous, that definitely includes that the peak. That's when the Earth has been the hottest it's ever been. Is in that particular period.
It's a PETM, yeah.
I know less about Cretaceous.
But there were others. During the late Cretaceous, there were other hypothermal events that might not have been as severe as the PETM, but were nonetheless hypothermal. So if they think that we've got a pulse of radiation occurring coincident with or because of potentially the PETM, then maybe it would make sense that we also had other ones that were occurring with hypothermals earlier on.
That's kind of cool. Side note, I cover, when I do my undergraduate first year teaching, I talk about the Paleo-Eocene thermal maximum and PETM, and I talk about how it was the hottest the Earth has ever been, the most rapid temperature changes we've ever seen, and the fossil data to it, and then it's all interesting. And then I compare it with today's climate change, and the increasing in temperatures now is quicker than the Paleo-Eocene, and I see their little faces get quite crushed, and I feel a bit bad. I'm like, oh, you don't know the planet's dying.
I know.
I have to be the one to tell you that.
I used to teach exactly the same thing, and it's quite. And I used to get. I had an exercise where you can go on the IPCC. Is it the IPCC, the Intergovernmental Panel on Climate Change's website, and you can on their website, and you can model. You can look at their models for how the Earth is going to warm and how local areas are going to change as a result of various different emission scenarios with different climate models. And you can actually go on the website and play around with it. And I used to do a practical where I used to get them to do this, and they could see what would happen to southern England or what would happen to Europe or different places. It's a super cool website. You can model it. But yeah, I mean, they come away absolutely. I mean, it's terrifying when you look at the stuff. Yeah. It is completely terrifying. That is a side note. Sorry.
Side note, the world's dying. Let's move on. Let's start talking about fossils again. So that was the end of their paper, basically. They got these two main pulses. I mean, I'm a bit frustrated that it doesn't contain any fossil data because, of course, we're only looking at one side of the picture there. There's a lot of things in the Cretaceous that angiosperms that aren't counted for here. Now, I guess Cretaceous pollen might be a bit more tricky to work with, but I think if we're going to tell this story, we definitely need that as part of the story.
It's anyway, a bit surprising. It's a bit surprising that they didn't look at the fossil record. I mean, maybe they did and say, oh yeah, these pulses correspond with the evolution of X or Y or something like that. Like
they did do a bit of that. They talked about how the pollen pulses seemingly correlate with changes in the vein density on leaves. So again, you need to refer to a different study with a different taphonomic. This is the problem we were referring to earlier.
Yeah.
When you're doing plants, you've got to refer to the two different studies. They do also refer to a study of fossil pollen But once again, that fossil pollen study is from 1997, 1998, something like that. So I was a bit like, what's going on
Bananas, yeah.
here? The world of plants. Well, not bananas, because they're going to go extinct. But yeah.
Oh yeah, sorry, not bananas.
Sorry. So this raises all sorts of interesting questions about when these things got going, about whole genome duplications are also relevant, because that's what plants do a lot of, especially in the context of the massive environmental change at the end Cretaceous. And also insects is also one of the things you might think about. When you think about pollen, what the insects are up to is really relevant, right? So if you're relying on some sort of insect pollination, what your morphology of your pollen is going to be really important about what the insects are doing and how they're using or working together. So, do you want another final plant afterthought?
Go on then, a plant afterthought. A
This
one-liner, Rob.
One-liner. This is another paper that came out in June 2026. So this is one of the reasons I didn't send it to you, because it's only just come out. This is Integrated Analysis of Fossils and Molecular Divergence Time Estimates, a latest Jurassic origin of angiosperms. So this is by Roisin Wu and colleagues from Bristol.
Okay, hang on. I need to find the right paper.
I haven't sent it to you. You don't even have it.
Oh. What have you sent me then?
I haven't sent you. You have to just go on this without. So
they've tried
to. They're raising they're trying to test this problem that there is a gap between what the fossil record says and what the molecular clocks say. So the molecular clocks say that angiosperms should have got going in the Jurassic and Triassic, but the fossil record is always much different. So they used a different kind of analysis. They've done a Bayesian-Brownian bridge model. So I was trying to get a handle on what this is. I should know what this is really, but they're trying to estimate the fossil divergences before they start putting them into the molecular clocks. This is without using a tree, just looking at the fossils and using all sorts of complicated analysis, looking at the stratigraphic fossil occurrences, and then they do an phylogeny and they estimate that using different kinds of Bayesian models, and they estimate a late Jurassic origin for crown group angiosperms, which dramatically diminishes the perceived Jurassic gap between the molecular clocks and the oldest unequivocal crown group angiosperm fossils. So that's kind of fun. They are using a different kind of technique to indicate that gap might not actually be real if you try and use different techniques.
Yeah, I think that people have suggested though that there are late Jurassic angiosperms. I mean, I'm sure I read a paper a couple of years ago about late Jurassic angiosperms from China, but maybe they're more controversial. Maybe they're not crown group, or maybe they feel that they're much more controversial or something like that. I don't know.
I'm afraid I don't know. This is the difficulty of me trying to choose to talk to you about plants. I should stay in my lane and choose animals next time, shouldn't I? But I think together this tells an interesting story, right? When did angiosperms get going? This is a problem that Darwin's been annoyed about from the very beginning. And actually, since Darwin, we've got a lot more data, but the fundamentals of the problem remain the same. Like, when did this massively important, diverse group of plants get going? And we've had a look. We can see that the fruits were actually quite big in the Cretaceous. Maybe the dinosaurs and mammals were eating them and dispersing them, and that the pollen was getting diversified and much more disparate and complex in the middle of the Cretaceous, not just waiting until the end Cretaceous event. So there's all sorts of cryptic things going on there. So that was my little plant story for you. I hope you don't mind me dragging you into the world of plants.
No, world of plants, very interesting. Like it a lot.
What do you think our take-home messages are going to be?
I
think we need systematic collection. I think maybe we need plants need more attention. I guess is one of the things we need. I need systematic collection of the data, not just chucking it in every now and again.
Agreed. I'm just reading. I'm just Googling now, reading a paper here. It was in eLife. Life. I mean, this is an MPDI journal in 2023. New fossil evidence suggests that angiosperms flourished in the Middle Jurassic. So this was a study. Early Jurassic Nanjing anthus, which is based on over 200 specimens of fossil flowers. So anyway, I guess this stuff is just controversial, and that I mean there are, but basically what I'm saying is there are fossils that people have suggested are angiosperms from the Middle Jurassic.
Well, if we take the combined data from those three papers we just talked about, this indicates that we should be looking at the late Jurassic for more plants, right?
I mean, the Jurassic is definitely, maybe this is going to be my take home. The Jurassic is the most interesting time to answer all these questions, and we just need to be working on the Jurassic, you guys.
Do you think we maybe need to fund people as they go and do Jurassic fieldwork in places that might have been underexplored, potentially, Susie?
I think that's an excellent thing to do, Rob. I think that would be a really good idea. Yeah.
I'll just send my emails now to the European Research Council saying fund Maitland et al. Now for an integrated study of Kyrgyzstan. Where was it you were going?
Morocco, yeah.
Kyrgyzstan, other places.
Yeah.
Yeah.
UK.
Just give, or just short version, give Susie money. Right. That's the answer to that question.
Thanks, Rob.
Right. And on that, that's a good place to leave it. I'll see you soon, Susie. Thank you.
See you later, Rob. Bye-bye.
Bye.
The Fossil Files was created by Robert Sansom and Susanna Maidman.
Fossil Files is a labour of love created in our spare time with no budget, so we really welcome any support you can provide us via our Patreon.
There you can access bonus content and join in the discussion by posting your own questions or even participate in live episodes.
In either case, we want to hear what you think. You can let us know what you thought about the topics we discussed or make suggestions for future papers in our social media channels.
You can find links to our Blue Sky, Instagram and YouTube channels on our website fossils.libsyn.com.